BCE Chemistry, Unit 4, and welcome to AOS 2. Let's start with proteins. Okay. Proteins are made up of amino acids. Okay. And amino acid is that cool acid that can act as a base. Okay. So what do you call an acid with an attitude? Never thought about that, huh? Amino acid. So kids, hide your lunch money because amino acid is here. Okay. So amino acids are building blocks of protein. Let's talk about protein first. Okay. So proteins are important to the body. Okay. The cell structure, such as muscles, are made up by proteins. Okay. So have a look at your bicep. If you can actually see them, you know the power of protein. If you cannot see them, then you know you need the power of protein. Okay. All hormones, cell membranes, transport molecules, and antibodies, they're all actually made up of protein. Okay. And what are proteins made up of? Amino acids. So amino acids are like the small molecules. They're actually the monomers that makes up a peptide chain. We'll talk about peptide later. And then that peptide chain curls and coils, and different peptide chains, curls and coils combined together makes up a protein. So protein is a really, really large molecule in chemical terms. Amino acids. So we said it's a monomer that's made up of, that actually makes up protein. And then there are 20 different amino acids that makes up the proteins in our body. Vika actually did us a favor to give us all the structures and names and acronyms of the 20 amino acids in our body on the data book. Go have a look. And if you have a look, then you will see that all of them are actually 2 amino acids. See here, regardless of the side chain, like the main chain is supposed to be actually go all the way here, according to like IUPAC naming, the longest hydrocarbon chain, right? But all of the way that they listed it, they really stresses the basic structure that they have in common, which is the amine, the amino functional group, is attached to the second carbon, which we call it alpha carbon, because it's the first carbon next to the cool group. Okay, so the amine is on the second carbon, which is right next to the cool group, and the cool itself, the carbon itself, cannot really have another group on top of it. So they are all 2 amino acids. So 9 of the 20 amino acids are called essential amino acids. They are the ones that our body cannot synthesize and must be obtained from food. And the other 11 is we can make, and hence they are not regarded as essential. So the amino acids always contain carbon, hydrogen, oxygen, and nitrogen. And sometimes the side chains might come with sulfur or phosphorus. So the amino acids used to synthesize these proteins are all 2 amino acids. So it is very important for us to know the structure that they all have in common. So we have the amino group here on one side, and then on the other side we have the carboxyl group. This is the number 2 carbon. It's the first carbon next to the carboxyl group. That's the alpha carbon because it's 1 and 2. Okay, and then you have the side chain. The side chain can be quite versatile. It can be polar, it can be non-polar, it can be proton donator or proton acceptor. Essentially it's saying that it can be anything. Okay, well, since amino acids, all the amino acids have the same basic structure here. Okay, so they only differ by the side chain, and in the side chain that actually determines their behavior and their properties and their functions essentially in our body. some people might have noticed that say for these amino acids, this carbon, this alpha carbon, is actually a chiral carbon because it's connected to 4 different groups. True. Actually, for most of the amino acids, actually 19 out of the 20 amino acids, that carbon, at least that carbon is 1 chiral carbon in the amino acids. So they're all chiral molecules, right? Some might even have more than 1 chiral carbons, okay? Except for one amino acid, that is glycine. Glycine is the simplest amino acid in our data book because it doesn't really have any side chains. It just have another H on the other side in the place of side chain. So glycine is not a chiral molecule, but rest of the amino acids, they're all chiral molecules. Based on the common structure of amino acids, do you think they are acids or base? Well, let's have a look. The amine part are proton acceptors. Okay, so they act as a base to receive proton, so it can actually receive a hydrogen cation and become positively charged. Okay, so notice where I have put the positive sign. It has to be on the nitrogen. So if you are asked to label this in a particular diagram, how to draw after it has received that hydrogen, how does it look like? Your positive charge also has to be added on N, okay? And then you have the acid part that are proton donors, okay? They donate hydrogen to the solution to become just a group without the H, okay? So both processes enable amino acids to form hydrogen bond with water, and therefore amino acids are soluble, and that's how they are transported in bloodstreams. Okay. So as a result, an amino acid in the molecule, an aqueous molecule, aqueous solution, can be in the form where it has one end that has a positive charge and the other end has a negative charge. But overall, the molecule doesn't really have a charge. So for molecules like that, we call it a sweater ion, or we call it a dipolar ion. Okay, it has a positive charge and a negative charge at the same time, but overall, it doesn't have a charge because it sort of like neutralizes. Okay. Well, but when the pH of the solution changes, this sweater ion, the dipolar ion, will also change with it. So if the pH reduces, becomes smaller, it means that the environment has increased in the concentration of hydrogen cations. Then the Q part, the COO part, because it has lost one hydrogen in a normal solution, now it has gained that hydrogen back in a reaction that looks like this. Okay, it's a reversible reaction. It really depends on the hydrogen cation in the environment. The lower the pH, the more the reaction will towards the right-hand side. Okay, so as a result, the sweater ion becomes a cation with a positive charge. Also, you can imagine that when pH increases, as in there are a lot of hydroxide around and that can accept protons, then the NH3 group can lose its hydrogen that was gained to the solution, to the hydroxide to make a water, and itself goes back to the NH2 group. So leaving the Q with a negative charge. So a sweater ion in the high pH becomes an anion.